Biochemistry 401 Lecture 30. Today we're going to talk about cholesterol transport. We're going to talk about the origin, function, and fate of lipoprotein particles, good cholesterol versus bad cholesterol, and then we're going to talk about lipoprotein particles and disease, including atherosclerosis and metabolic disorder. So let's get started. Lipoprotein particles are comprised of both lipids and proteins, just as the name suggests. The lipids include phospholipids, triglycerols, cholesterols, and cholesterol esters, and other lipids, such as lipid-soluble vitamins. The proteins are called apolipoproteins, and sometimes they're called apoproteins. Apolipoproteins serve many purposes. They help to organize the lipoprotein particle, and they help to target it to the right place. Some apolipoproteins also act as enzyme cofactors. Lipoprotein particles have a polar exterior that faces the aqueous solution and a hydrophobic interior. Lipoprotein particles are constructed such that the polar groups are situated facing the outside toward the aqueous solution, and the hydrophobic portion face the inside of the lipoprotein particle, and that's as you would expect. Cholesterol is oriented with the hydroxyl facing the outside, and the ring structure and long hydrocarbon tail pointing toward the inside. Here we see phosphatidylethanolamine. The long hydrocarbon tails are facing inward, and the ester linkage and phosphate alcohol head group are pointing toward the outside. The same thing is true for triacylglycerols. The fatty acids are pointing toward the inside of the lipoprotein particle, and the ester linkages are pointing toward the outside. This makes the ester linkages accessible to lipases. This is important in the hydrolysis of triacylglycerols. Lipoprotein particles also contain apolipoproteins, and these are also found toward the outside. And packed in the center of lipoprotein particles, we find cholesterol esters. This is cholesterol that has been acylated. A fatty acid has been added at the hydroxyl so that it can pack more tightly into the center of the lipoprotein particle. Plasma lipoproteins were originally isolated and characterized based on their relative densities. Plasma lipoproteins are shown in the figure below and are listed in this chart. The name of the plasma lipoprotein is shown first, then its density, its diameter, the type of apolipoprotein it contains, its physiologic role in the body, and its percent composition of triacylglycerols, cholesterol esters, cholesterol, phospholipids, and protein. We're going to start with chylomicrons. These are the least dense, and they have the highest diameter. They contain apolipoprotein B48 and C and E. Chylomicrons primarily contain triacylglycerols of dietary origin, and therefore the physiological role of chylomicrons is to transport fats. The rest of these plasma lipoproteins are constructed in the liver. They range from very low density lipoprotein, which are the least dense of those that are generated in the liver, all the way down to high density lipoprotein particles. They vary not only in their density and diameter, but also in their physiological role. For instance, high density lipoprotein is what you may have heard referred to as good cholesterol. This is a lipoprotein particle that is engaged in reverse cholesterol transport. High-density lipoproteins take cholesterol from the tissues and bring it back to the liver. The rest of these, the very low-density lipoproteins, intermediate-density lipoproteins, and low-density lipoproteins, all carry endogenous lipids from the liver to the tissues. Very low-density lipoproteins are constructed in the liver, and like chylomicrons, they primarily contain triacylglycerols. Their role is to carry endogenous fat in the body and to transport it to the tissues, primarily the muscles and the adipose tissue. And as more and more triacylglycerols are given off, very low-density lipoproteins become intermediate-density lipoprotein particles. As they give off more of their lipids, they become smaller and therefore more dense. Intermediate density lipoproteins are then turned into low density lipoprotein particles, and these are primarily involved in cholesterol transport. They primarily contain cholesterol esters, and so low density lipoprotein is known as the bad cholesterol. It is involved in transport of cholesterol originally from the liver to the tissues. Now, the apolipoproteins that are found in chylomicrons differ from those that are found in lipoprotein particles that arise in the liver. Chylomicrons contain B48, for instance. Those lipoprotein particles that contain endogenous lipids contain B100 apolipoprotein. And so what do you need to know about this chart? There's a lot of information. Well, you need to know the names of the plasma lipoproteins and whether they're larger or smaller in comparison to one another, and whether they're more dense or less dense in comparison with each other. You also need to know what sort of B apolipoprotein they contain, and which component has the highest percentage composition in each lipoprotein particle. For instance, in chylomicrons and in very low density lipoproteins, the component with the highest percentage are triacylglycerols. In intermediate density lipoprotein, it's about a 50-50 split between triacylglycerols and cholesterol esters. Whereas in low density lipoprotein particles, this is cholesterol ester. And in high density lipoprotein, it's protein. And that's it. Please do not memorize these numbers. You do not have to memorize the numbers. I hope this helps. So why is it that chylomicrons, which are formed in the small intestines, have a different apolipoprotein B than those lipoprotein particles that are constructed in the liver? The small intestine contains an enzyme, a deaminase, that removes the amino group in cytosine to form uracil. This introduces a stop codon into the messenger RNA. Therefore, the ribosome stops short and translates a shorter B apolipoprotein. In fact, it's only about half as long as ApoB100. This is an intestinal-specific apolipoprotein, and it's only found in chylomicrons. Now, chylomicrons, as we said, are formed in the small intestines, and they have 3 stages of development. They start off as a nascent chylomicron. They have all their lipids, but they don't have all their proteins, and we'll talk about that in just a minute, and therefore they're not functional. These nascent chylomicrons are found in the intestines and in the lymphatic vessels, and are found early on in the bloodstream. In the bloodstream, chylomicrons become mature chylomicrons. This is due to the addition of apolipoprotein C2. Once they give off the majority of their triacylglycerols through hydrolysis, they become known as chylomicron remnants. These are the leftovers, and they're returned to the liver for recycling. Let's take a look at this. Here we see epithelial cells of the intestine. These are enterocytes. In the enterocytes, apolipoproteins and lipids come together to form chylomicrons. These consist of triacylglycerols, cholesterol esters, phospholipids, and other lipids. And they contain ApoB48, as we just saw, and another apoprotein, ApoA1. These are constructed, and then they enter the lymphatic vessels that underlie the epithelial cells. They travel in the lymphatic system and enter the bloodstream in a place close to the neck, believe it or not. And these chylomicrons, as they travel in the lymphatic system and as they just enter the bloodstream, are nascent chylomicrons. They're not functional yet. But in comes HDL, good cholesterol, and it's going to take ApoA1 from the chylomicron, and in exchange is going to transfer ApoC2 and ApoE to the chylomicron. And now, because the chylomicron has the apolipoprotein ApoC2, it is an active and functional chylomicron. This is known as a mature circulating chylomicron, and it's going to deliver triacylglycerols and other lipids to the tissues, primarily to adipose tissue and to the muscles. And the way this happens is in association with a lipase that is found on the endothelial cells of capillaries that surround these tissues. Once they have delivered their triacylglycerols, they actually lose their C2, and they're known as chylomicron remnants. They go back to the liver, bind to receptors on liver cells, and are endocytosed into the liver cells. So now let's talk about how things happen. We're going to talk about how triacylglycerols are delivered from the lipoprotein particles to the peripheral tissues. Now we're going to go over what happens with chylomicrons, but similar things happen with VLDLs also. It's the source of the triacylglycerols that's different. With chylomicrons, they're primarily of dietary origin, and with VLDLs, they're primarily of endogenous origin. So let's see how this happens. Here we see a membrane of an endothelial cell that overlies the adipose and muscle cells. This is a cell that lines a capillary. Now on the surface of the endothelial cell is an enzyme that's called lipoprotein lipase. This is going to be activated by ApoC2 from a chylomicron or from a VLDL. They both have ApoC2, and ApoC2 activates lipoprotein lipase. Triacylglycerols are going to be hydrolyzed through the action of lipoprotein lipase, first to diacylglycerol and a free fatty acid, and then to monoacylglycerol and a fatty acid. And these are transported first into the endothelial cells of the capillaries, and then they're transported to the adipose tissue and the muscle cells, and other peripheral tissues as well, but primarily adipose and muscle tissues. We've just gone through the action of chylomicrons to release free fatty acids and monoacylglycerols to the adipose tissue and the muscle tissue to form chylomicron remnants that are then bound to remnant receptors and endocytosed into the liver, and the lipids and the proteins are recycled. We're now going to talk about the transport of endogenous lipids, VLDLs, IDLs, LDLs, and HDLs. VLDLs are formed in the liver, and just like chylomicrons, they take up apolipoprotein E and C2 from HDL. Now the VLDL is activated, and it's going to bind to the endothelial cells in the tissues. ApoC2 binds to lipoprotein lipase. The triacylglycerols are hydrolyzed to yield free fatty acids and monoacylglycerol, which are taken into the endothelial cells of the capillaries and then are transported to the tissues. Now, as these VLDLs give off the triacylglycerols, they become smaller and smaller, and in this way they're turned into intermediate density lipoproteins, IDLs, and then eventually low density lipoprotein particles, LDLs. These travel in the blood and bind to LDL receptors and are endocytosed. And the lipids found in these LDLs and IDLs are recycled for use by the body. Intermediate density lipoprotein particles and low density lipoprotein particles both bind to LDL receptors and are internalized into hepatocytes, liver cells, through endocytosis. And so these lipoprotein particles bind to the LDL receptor, as shown in panel A, and then they're endocytosed, brought into the hepatocyte, and this is shown in panel B. This is the pathway for endocytosis of lipoprotein particles. We're going to start at 1 and we're going to end at 6A and B. Let's walk around this diagram. In this diagram, the lipoprotein particles are shown as yellow spheres, and the B apoproteins are shown as red squares. The LDL receptors are shown as dark blue rectangles, and the coating that's found at the membrane shown in magenta. This is a protein coating of the membrane that helps to make an endocytic vesicle. So let's start at 1. Here we see a lipoprotein particle that's ready to bind to the LDL receptor. At number 2, the LDL or the IDL has bound to the LDL receptor, and in number 3, this part of the membrane has pinched off from the cell surface. In number 4, we see that the vesicle has now been uncoated. The protein coat has left. This vesicle now becomes acidified to form a lysosome. This is shown in number 5. In the lysosome, because the vesicle has become so acidified, the LDL particle dissociates from its receptor. The apolipoprotein B is degraded by a lysosomal protease, and in 6A, the LDL receptors bud off and are recycled back to the membrane. In 6B, the cholesterol from the lipoprotein particles is released and it becomes esterified by the acyl-CoA acyltransferase enzyme, and we'll talk about this in just a minute. Now, cholesterol is esterified for two reasons. First of all, the presence of cholesterol in membranes has an effect on the fluidity of that membrane, and we want to make sure that we have just the right amount of cholesterol in our membranes. Second of all, because an esterified cholesterol is more hydrophobic, it can pack more tightly for storage. Here we see the reaction by which cholesterol is esterified. We start with cholesterol plus an acyl-CoA. This is an activated fatty acid. And then the acyl group is transferred to cholesterol hydroxyl. This yields cholesterol ester and coenzyme A. The enzyme that catalyzes this reaction within the cells is called acyl-CoA cholesterol acyltransferase, abbreviated ACAT. Now, we haven't talked about the catabolism of cholesterol, and there's a reason for that. Cholesterol is really not catabolized in the way that we think of it. It's scavenged from the tissues by HDL, high-density lipoprotein particles, and then HDL travels to the liver and is endocytosed to let off that cholesterol. Now, some of the cholesterol is converted to bile salts, and as we saw before, that's an amphipathic derivative of cholesterol. It's synthesized in the liver, it's stored in the gallbladder, it's released into the small intestine, but 95% of the bile salts that are released into the small intestine are then taken up again by the small intestine, along with emulsified triglycerols and other lipids from the diet. Only 5% of the bile salts released are excreted in the feces. Think about this for a minute. This means that we have to be really careful in the synthesis of cholesterol and its utilization. The synthesis of bile salts requires an enzyme called cytochrome P450. This is a heme-containing monooxygenase enzyme. This is an enzyme that catalyzes the addition of one oxygen to oxygenate sterols like cholesterol. It's important in the synthesis of bile salts and also in steroid hormone synthesis. Cytochrome P450 is also used in detoxification, and so it's important to regulate the amount of unesterified cholesterol in the cell. High levels of unesterified cholesterol in the cell have three major effects. First of all, as we saw before, is the inhibition of HMG-CoA reductase. HMG-CoA reductase catalyzes the rate-limiting step in cholesterol synthesis, and as we saw before, it is highly regulated. The second thing is the activation of ACAT. This increases the esterification of cholesterol so that it can be stored, which is much more safe. Third is the inhibition of LDL receptor synthesis to reduce the amount of LDL receptors that are on the cell surface and thereby reduce the intake of LDL particles from the blood. And the last one can be a bit of a problem. So this brings us to the good, the bad, and the ugly. We're going to talk about good cholesterol and bad cholesterol and the role of lipoprotein particles in coronary heart disease and metabolic syndrome. LDL receptors, we said, are required for IDL and LDL uptake. Now, normally, about half of the intermediate density lipoprotein particles are returned to the liver via LDL receptor-mediated endocytosis. The rest of those IDLs become LDLs. The amount of circulating LDLs, therefore, depends in large part on how well IDL can be removed from circulation. And this, in turn, depends on the number of LDL receptors that are found on the cell surface relative to the amount of circulating IDL particles. It also depends on the proper functioning of the LDL receptors themselves and the process of receptor-mediated endocytosis as a whole. Those IDLs that can't effectively get into the liver cell remain in circulation, and as they give off their lipids, become smaller and smaller to become LDLs. Those LDLs that can't get into the liver cells by endocytosis also remain in circulation. They give off their lipids and they become smaller and smaller as well. This is a problem, and we'll see that later. Now, when too much cholesterol is left in the blood, this is called hypercholesterolemia. There's too much LDL left circulating because it's not internalized at a sufficient rate. This can come from two main reasons. Either there's a problem with the LDL receptor, or long-term ingestion of dietary cholesterol that far exceeds normal requirements. One of the main causes of hypercholesterolemia is a defect in the LDL receptor. This is referred to as familial hypercholesterolemia, FH. Now, when there's a defect in both copies of the gene that gives rise to the LDL receptor, the serum cholesterol is 3 to 5 times normal levels, and this begins in childhood. A person heterozygous for this deficiency has a serum cholesterol that's about twice normal levels by middle age. Now, in each case, there are not enough LDL receptors on the surface, or there are a normal amount of LDL receptors, but they just don't work very well. They don't bind to the lipoprotein particles very well, or they don't internalize them very well. And the second way is when there's long-term ingestion of dietary cholesterol that far exceeds daily requirements. This means that too many LDLs get into the cell. And as we saw, when there's non-esterified cholesterol in the cell, this results in an inhibition of LDL receptor synthesis. So with high cholesterol, we have a decrease in LDL receptor synthesis, whereas with low cholesterol, there's an increase in LDL receptor synthesis. So with long-term ingestion of dietary cholesterol, it's thought that this decreases the amount of LDL receptors that are available on the cell surface, and therefore too much cholesterol remains in the blood. Now, does this dog look like trouble? I had a lab one time that was a holy mess. You'd leave her alone and she would tear the house up. She would go through all my potted plants, she'd rip up all my shoes, the whole business, just from top to bottom. Damage, damage, damage. That's the trouble that one lab can cause. Can you imagine if you had a whole bunch of them that were all left alone with nothing to do for hours and hours and hours? More trouble. This is the same thing with too much LDL left roaming around in the bloodstream. If they can't get where they're supposed to, if they can't bind to the LDL receptor and be internalized the right way via endocytosis, those LDLs will find another way. Those LDLs will get into a lot of trouble. Let's look at this. And so, when there are too many LDLs left in circulation, they will release more and more lipids to become smaller and smaller. These small LDLs are very dangerous because they can actually fit in between the cells of the endothelium and go right into the tissue of the blood vessel itself. Once there, the components of the LDL particle can become oxidized, and this results in inflammation. This inflammation recruits white blood cells to the site. Monocytes come and they go between the endothelial cells. There they become transformed into macrophages and release cytokines to attract more and more white blood cells to this site. The macrophage is called a macrophage because it's a big eater. It ingests foreign particles as part of our defense system against invading organisms. And here the macrophages ingest the LDL components to become what's called foam cells. They're called foam cells because of the presence of so much lipid in the cell. This high lipid content gives the foam cell a bubbly, foamy appearance. Foam cells secrete growth factors and metalloproteinases that degrade the blood vessel itself and cause scarring. This scarring reduces the flexibility of the blood vessel, leading to what's called a hardening of the artery, atherosclerosis. The blood vessel is becoming scarred, which is also called sclerotic. Blood vessels that are scarred this way have a harder time increasing and decreasing their diameter, and this leads to big problems. And so here we see the progression of atherosclerosis. This is just a recap, and there are notes that you can use to study. As more and more lipids build up in the blood vessels, this can cause the formation of something called a fatty streak, because that's what it looks like in the blood vessel itself. Now, fatty streaks can get larger and larger and can become calcified to become plaques. These plaques can rupture and cause blood clots to form. These blood clots, called thromboses, can cause an infarction, which is a blocking of the blood vessel. Now, if blood vessels in the heart are blocked, it can result in a heart attack. If blood vessels leading to the brain are blocked, this can lead to a stroke. In this figure in panel A, we see a normal blood vessel with a clear, large lumen through which blood can flow easily. In B, and in the panel on the far right, we see blood vessels in which the lumen has been severely constricted by the presence of large lipid deposits, which fills most of the blood vessel itself. This plaque is circled in red, and there's an arrow pointing to it. You can see how small the diameter of the artery has become. Now that's bad cholesterol. LDL is bad cholesterol when there are too many of them and they're too small, because they can get into bad places and do bad things. HDL is called good cholesterol because it has protective effects. It is involved in the reverse transport of cholesterol from circulating lipoprotein particles, the reverse transport of cholesterol from peripheral tissues, and as we saw, the transfer of components to and from other lipoprotein particles. HDLs take excess cholesterol from the peripheral tissues and from other lipoprotein particles and bring it to the liver. There it can be stored, it can be recirculated, or it can be made into bile salts and released into the small intestine. There's called a double-pronged approach to cholesterol reduction, and this involves attacking the problem two ways. First of all, by inhibition of synthesis itself by statins like lovastatin, and secondly, by inhibiting the reabsorption of bile-associated micelles, and this prevents their uptake, and instead, these micelles, which include bile salts, cholesterol, fatty acids, triglycerols, lipid vitamins, are kept in the intestines and are eliminated in the feces. Now, statins are competitive inhibitors of HMG-CoA reductase, and as you'll recall, this catalyzes the rate-limiting step in cholesterol synthesis. On the left, we see 3-hydroxy-3-methylglutaryl-CoA, HMG-CoA, and this is the substrate for HMG-CoA reductase. Lovastatin looks enough like HMG-CoA that it binds to the active site and inhibits HMG-CoA reductase. So that's what statins do. They're competitive inhibitors of HMG-CoA reductase, and some of the current statin drugs are shown here. Do you need to know all the names? Oh, no, you sure don't. But you do need to know what they do and how they do it. Now, too much cholesterol in the blood can lead to coronary heart disease. Another thing that can lead to coronary heart disease is something called metabolic syndrome. This can lead to both coronary heart disease and to type 2 diabetes. Let's look at this a little more closely. The diagnosis for metabolic syndrome is established when more than 3 risk factors that are shown below are present. This includes a large waist circumference, high triglycerols in the blood, low serum HDL, high blood pressure, and a higher than normal fasting glucose. Do you have to memorize the numbers that are shown in the table? No, you don't. Not right now anyway. Not for this class. You do need to know the risk factors, though, and how diagnosis of metabolic syndrome is tied into these risk factors. Metabolic syndrome is a big problem. not only because it can lead to coronary heart disease, but also because it can lead to type 2 diabetes. And so the NIH National Heart, Lung, and Blood Institute conducted a study, and the study found out that exercise and diet change that resulted in significant weight loss had a pronounced effect on the reduction of progression from metabolic syndrome to type 2 diabetes. Now, type 2 diabetes is often called adult onset diabetes because this is generally when the symptoms first present themselves. However, more and more these days, this type 2 diabetes is also seen in children, and this is a big problem. So I'm going to go through this really quickly, and you do not need to know the numbers that are presented here. You just need to know the concept. 3,234 men and women that had impaired glucose tolerance, this is a higher than normal fasting glucose level, were randomized in 3 groups. The first group undertook lifestyle changes. This included at least a 7% weight loss, and they exercised 150 minutes a week. The second group took a drug called metformin, and the third group took placebo, a sugar pill. And they looked to see who developed diabetes and who didn't. This was supposed to be a 3.5 to 5 year study, but it was discontinued about a year early because the results were conclusive. They had their results. The persons who took a placebo didn't lose very much weight at all. Not in 6 months, not in 12 months, not in 4 years. With metformin, there was an initial loss of weight for the first year, but then there was a gradual increase. And although these people lost some weight, they didn't lose as much as those who undertook a lifestyle change. For these people, they had a dramatic loss of weight in 6 months, and gradually they put some more weight back on. But still, 4 years later, these people continued to have a lesser weight than those either on placebo or on drugs. And so even better, when they looked at the incidence of type 2 diabetes in these 3 groups, 1 year, 2 years, 3 years, and 4 years after the start of the program, these researchers looked at the cumulative incidence of diabetes as a percent. 40% of those on placebo who initially had metabolic disorder went on to develop type 2 diabetes. For those who took metformin, this number decreased by 31%. For those who undertook an intensive lifestyle modification through reducing body weight and increased activity, had an incidence of progression to type 2 diabetes that was 58% lower than those who were on placebo. And so this study shows quite clearly that having a healthy lifestyle, watching what you eat, and getting enough exercise matters. This is the end of Lecture 30. Have a great day.